Hello, and welcome to my poster presentation, Target-Related Alpha Attenuation in a Brain-Computer Interface Rapid Serial Visual Presentation Calibration Task. My name is Dan Klee, and I'm a research associate in the Department of Neurology at Oregon Health and Science University in Portland, Oregon. My collaborators are all members of the Consortium for Accessible Multimodal Brain-Body Interfaces, or CAMBI. The work presented today examines the viability of using posterior alpha rhythms in a particular type of non-invasive brain-computer interface, or BCI. BCI systems evaluate brain signals with the general goal of assisting a user to perform movement or communication-related tasks. One variety of non-invasive BCI is the Rapid Serial Visual Presentation Keyboard, or RSVP, which is a P3 speller paradigm that utilizes event-related potentials, or ERPs, to differentiate target and non-target brain responses. Though P3 spellers are a popular BCI methodology, posterior alpha rhythms are another well-studied brain signal known to index aspects of visual selective attention. Of note, the widely documented attenuation of posterior alpha amplitude in response to target visual stimuli is not commonly implemented in BCI systems. As such, we performed an experiment to test several questions. Are there measurable target-related decreases in posterior alpha amplitude during the BCI-Pi RSVP task? If so, do these attention effects change when varying presentation rate? To what extent do any observed alpha attenuation effects correlate with coincident evoked N2 and P3 potentials? And lastly, can we use alpha amplitude changes within RSVP to classify target and non-target stimulus responses? In this experiment, 12 generally healthy adults completed 2 instances of RSVP calibration at 1 and 4 Hz presentation rates. EEG data were collected using a wearable sensing VR300 dry electrode system. All data were filtered and downsampled prior to continuous wavelet transforms and ERP processing. Statistical tests of time frequency data were conducted using the wavelet layer closest to each individual participant's peak alpha frequency. Each RSVP calibration task consisted of 100 sequences with 10 letters presented per sequence. Participants viewed a target prompt for 1 second, followed by a fixation for 500 milliseconds, and then 10 randomly ordered letters composed of 1 target and 9 non-targets. As mentioned, the presentation rate of these letters was either 1 Hz or 4 Hz. So, what did we find? Heat maps of mean frequency content 4 to 20 Hz are shown separated by stimulus type and presentation rate. Center lines indicate stimulus onset, and the black boxes show alpha range in the effect window of 300 to 800 milliseconds post-stimulus onset. Plots show a clear alpha desynchronization in the 1 Hz condition and a similar, albeit much fainter pattern at 4 Hz. Across participants, there was a highly significant reduction in alpha amplitude z-score following targets relative to non-targets. This attention effect was present at 4 Hz as well, but significantly blunted. Within participants, these effects were somewhat more variable. Half of participants showed significant alpha attention effects at 1 Hz, but only 1/3 demonstrated effects at 4 Hz. ERP analysis yielded significant target-related N2 and P3 attention effects. N2 amplitude increased in size from the 1 Hz to the 4 Hz condition, while P3 amplitude decreased. To compare alpha and ERP attention effects, we plotted target-non-target differences in mean alpha amplitude against changes in N2 and P3 as indexed by difference wave amplitudes. There were no significant correlations between these measures. Finally, we assessed classifier performance. RDA KDE bars at the far left represent BCI-Pi's default ERP AUC estimates, while the next set of blue bars demonstrate AU estimates from SPSS based on mean alpha activity in the effect window. The remaining 7 categories offer results of alpha amplitude time series data fed into a variety of other models, including a uniform random approach for the sake of comparison. Alpha classification AUC of target versus non-target stimuli was quite low compared to the standard ERP RDA KDE estimates. ERP classification went up between the 1 and 4 Hz conditions, while all alpha models, sans multilayer perceptron, demonstrated significant decreases. None of the alpha classifiers correlated with the ERP classifier. And so, to summarize, alpha attention-related attenuation is indeed measurable in the RSVP task, both across and within individuals. This effect is ostensibly unrelated to attentional ERPs in the same task, though classification of these alpha signals is low compared to these same ERPs. One possibility is that the analysis windows we use need to be modified on a case-by-case basis when measuring alpha within RSVP. Although alpha classifiers underperformed relative to the ERP classifiers, they were in most cases above random chance. Lastly, there is evidence against the redundancy of using both ERP and alpha analysis to bolster system performance. Thank you so much. Again, my name is Dan Klee, and I can be reached at klee@ohsu.edu. I'd like to thank all of my fellow researchers at CAMBI. Please feel free to reach out via email as shown on the screen. Thank you.